Hello students, welcome to the EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today I am going to talk about the classification of lipids from the paper Lipid Metabolism. The objectives of this paper includes the classification of lipids, to understand it, how many classes of lipids are present, and what are the significance of each and every class of lipids. As far as lipids are concerned, classification wise, there may be saponifiable lipids and non-saponifiable lipids. The saponifiable lipids has one or more ester groups, allocating it to endure hydrolysis in the charisma of an acid, base, or an enzyme. On the other hand, the non-saponifiable lipids include the phospholipids, the prostaglandins, the sphingolipids, the steroids, the terpenes, the triglycerides, waxes, which cannot be raked up into smaller molecules by hydrolysis. So this is one way of classifying the lipids. Again, based on the polarity, the lipids are divided into two basic classes: the polar lipids as well as the non-polar lipids. According to Kohn and Stumpf, who gave the classification of lipids in 1966, lipids can be classified into four different types. The first is the natural lipids. The second, the phosphorus containing lipids or the phospholipids. Third category includes the carbohydrate or the saccharide containing lipids, which is called as glycolipids. And the fourth category includes the terpenes or the terpenoids. The phospholipids and the glycolipids can further be sub-classified into different types, which we come to study in the later slides. After Kohn and Stumpf in 1966, Davidson et al. 1977 gave the triangular classification of lipids. He described lipids as energy storage lipids, lipids containing phosphorus group or the phospholipids, and the steroids, which contains cholesterol, and this classification is different from that of Kohn and Stumpf. So as to understand the classification of lipids, the traditional classification includes the saponifiable lipids and non-saponifiable lipids, or we can say that lipids in which the saponification process takes place is saponifiable lipids, which includes both the simple lipids as well as the complex lipids, whereas the non-saponifiable lipids includes the derived lipids, which is further subclassified into terpenoids, carotenoids, quinones, and steroids. The saponifiable lipids contains fatty acids, which may either be saturated fatty acids or unsaturated fatty acids. Also, the simple saponifiable lipids may further be subdivided into glycerides, cerides, and stearides. On the other hand, the complex saponifiable lipids is further subclassified into phosphorus-containing complex saponifiable lipids, sulfur-containing complex saponifiable lipids, and lashing complex saponifiable lipids. These lipids react with sodium or potassium hydroxide to yield soap. The saponifiable lipids is having one or more ester groups, allocating it to endure the hydrolysis process either in the presence of an acid or a base or an enzyme. There are some structures of the saponifiable lipids as shown here, like glyceryl tristeariate, phosphatidylcholine, and constituent of beeswax. The non-saponifiable lipids, in which the process of saponification does not occur, includes the phospholipids or the phosphorus-containing lipids, prostaglandins, sphingolipids, steroids, terpenes, triglycerides, and waxes, which cannot be rigged up into smaller molecules by hydrolysis. The structure of cholesterol and prostaglandin PGE1 shown here represents the non-saponifiable lipids. On the basis of the polarity, the polar nature or the non-polar nature, the lipids are divided into two classes: the apolar lipids and the polar lipids. The apolar is also called as non-polar lipids, and the other category is the polar lipids. In polar lipids, the characteristics includes that it forms bilayer impulsively in water. Phosphatidylcholine restrain up to 30 combinations of fatty acids on its chain. The polar lipids exist in four states. The first, crystal state; second, the gel state; third, the liquid state; and fourth, the liquid crystal state. All these states are the peculiar characteristic of polar lipids. So beside these four states, the polar lipids also exhibits a sharp Tm, or the melting point, defined from the gel to liquid crystal transition. The Tm is very high for the saturated chains and very short for the polyunsaturated chains. In the liquid crystal state, biological membranes are always just over the Tm. The examples are fish. There are three subclasses of polar lipids, and these subclasses include anionic polar lipids, uncharged polar lipids, and zwitterionic polar lipids. However, no cationic lipids are reported in nature. So among the polar lipids, there are anionic lipids, and these anionic lipids are, for example, cardiolipins, phosphatidylglycerol, phosphatidylinositol, phosphatidylserine, and many others. There are some points which are peculiar to the anionic lipids, that they agree the X-ray crystallography structures with NMR conformations. The interaction by screening of cations from solution and hydrogen bonds. The pH at the surface lower than that of water, and the pH difference wanes off at high ionic strength. The anionic lipids constantly being involved in fusion, and surface can bind calcium and hydrogen, which eases the fusion process. The cardiolipins act as an acid anion, provokes hydrogen ion to operate as an attractant. The fusion arises because of the acid anion formation, and the negative charges become strikingly by trapping the protons. The entire membrane have a negative charge on their exterior that attracts the cations, for example, hydrogen ions and sodium ions. Similarly, all biological membranes have a proton gradient, which are sustained by a proton pump, except the presence of sodium ion gradient in animal plasma membrane. Some of the structures and the examples of the anionic lipids are shown in these slides. Cardiolipin, phosphatidylinositol, phosphatidylserine, and phosphatidylglycerol. The uncharged lipids example includes the surface of gangliosides, and sphingolipids are anionic but have hydrogen bond lateral interactions. Glycolipids have hydrogen bond amid neighbors, with each lipids having 3 neighbors. So the cerebrosides, the gangliosides, and the glycolipids are some of the examples. So among the polar lipids, there are deuterionic lipids, and the examples of the deuterionic lipids are phosphatidylcholine, phosphatidylethanolamine, sphingomyelin, etc. Some of the important features of the deuterionic lipids are they are position-wise. Phosphate is on the top of the glycerol, and glycerol is upright to the membrane. Second important point includes the primary ester is underneath the glycerol, and the secondary ester is parallel to the membrane before the chain plunges. Choline as well as ethanolamine gyrates at an angle of 15°C to the plane. Each head group of deuterionic lipids interact with three neighbor ionically. The deuterionic lipid examples include phosphatidylcholine, sphingomyelin, and phosphatidylethanolamine. The nonpolar lipids not spread to form a monolayer, insoluble in bulk, and the charge allocation is uniformly distributed. A very good example of this category includes the triglycerides. Further classification were based on the functional basis. Earlier, saponification was the characteristic for the classification of the lipids, followed by the polarity, and then function were assigned as the basis of classification of lipids. So looking at the functional basis, the lipids can be classified into storage lipids, structural lipids, as well as lipids that act as signaling cofactors and pigments. The storage lipids include the fatty acids, the triacylglycerols, and the sterols. The storage lipids is the reservoir of the energy. The main function of the storage lipids is to restore the energy and dissipate whenever and wherever it requires. The second category, the class which is assigned on the basis of function, includes structural lipids. And these structural lipids, especially present in membranes, are either containing phosphorus group or a carbohydrate or a saccharide. The phosphorus containing lipids are termed as phospholipids, whereas the saccharide or the carbohydrate containing lipids are the saccharolipids. The third class, which is assigned on the basis of function as signal, cofactor, and pigments, includes the lipoproteins, the proteolipids, as well as polyketides. All these are amalgamation of lipids with proteins. and they act in a signaling molecule as a signaling molecule in a signaling pathway or as a cofactor or as a pigment. The amphipathic nature of membrane lipids and the orientation of their hydrophilic as well as hydrophobic regions articulate their stuffing into the bilayer. Among the structural membrane lipids, three types have been described. The first one is glycerophospholipids, where the two fatty acid is connected to the glycerol present in the hydrophobic region. The second, sphingolipids, where the sphingosine, a fatty amine, is linked to a single fatty acid. And the third, sterols, the compounds with four fused hydrocarbon rings arranged in an inflexible arrangement. The glycerophospholipids and sphingospholipids include the charged alcohol at their polar end, and a few in addition contains the phosphate moiety. In these amphipathic compounds, the hydrophilic moieties could be more complex or as elementary as a single hydroxyl group at one end of the sterol ring system. Among these membrane lipids classes, colossal miscellany results from various combination of tail as well as polar head of fatty acids. Another way to classify lipids is based on hydrolytic product formation. The products of hydrolysis have also been considered for classifying the lipids into several categories. And these categories include the simple lipids, the complex lipids, as well as the derived lipids. The simple lipids are the lipids which, on hydrolysis, yields alcohol as well as fatty acids. The alcohols may be monohydric alcohols or trihydric alcohols. And similarly, in simple lipids, after hydrolysis, the fats or the lipids gets either identical fatty acids or dissimilar fatty acids. So the simple lipids may further be subdivided or subclassified into simple glyceride, mixed glyceride, oils, fats, and waxes. And the simple glycerides are the simple lipids that includes identical fatty acids. Here, in this subclass of simple lipids, the fatty acids are similar. Whereas in the mixed glyceride subclass of simple lipids, the fatty acids are of dissimilar nature. The oils, which are part of simple lipids, is the glycerol with unsaturated fatty acids. In similar lines, fats are the glycerols containing saturated fatty acids. And finally, the waxes subclass of simple lipids contain mono or dihydric alcohols with fatty acids. So, based on the hydrolytic product, the complex lipids is another class of lipids, also referred to as compound lipids or composite lipids. On hydrolysis process, it yields fatty acids, it yields glycerol, phosphoric acid, along with serine, sphingosine, ethanolamine, and various sugars. The complex lipid class can be further subdivided into phospholipids, where cephalins, glycolipids, which further be subclassified. The phospholipid subclass of the complex lipid includes the glycerol moiety, the fatty acid moiety, the phosphoric acid, as well as the nitrogenous base. The example include lecithin, where a glycerol moiety, a fatty acid moiety, a phosphoric acid, as well as choline is present. Cephalin, another subclass of complex lipid. It includes glycerol moiety, fatty acid moiety, phosphoric acid moiety, as well as ethanolamine. The glycolipid subclass of complex lipid contains glycerol moiety, fatty acid moiety, as well as carbohydrates. And complex lipids can be further subclassified as sulfolipids, galactosyl diglyceride, and cerebrosides. The sphingospholipids is another subclass of complex lipid, where sphingosine moiety in place of glycerol is present. Fatty acid, phosphoric acid, and choline is present. So the difference between the sphingophospholipid and phospholipid is the presence of sphingosine. The third hydrolytic base class of lipids are derived lipids, and these derived lipids are the hydrolytic commodities of complex and simple lipids. Examples are alcohols, which includes sterols and glycerol, fatty acids and terpenoids, etc. So the three different classification of lipids have been described. So today we have learned the classification of lipids. There are many ways people have defined the classes. If we look at the Kohn and Stumpf classification of 1966, it was described that there are four classes of lipids. The first one is the natural lipids, the second, which is phospholipids, the phosphate bearing lipids, the third is the glycolipids, where carbohydrate is the moiety, and finally the fourth group, which is the terpenoids. So lipids are classified in other numerous ways. Say for example, look at Davidson et al. Classification, which classify lipids into energy storage lipids, phospholipids, as well as steroids. So, in this lecture, we have learned about the different types of classification of lipids and this incredibility of the properties and functions classify the lipids. Thank you.